welcome everyone. Thanks for joining us today for the Tools and Technology Seminar Series. I have one quick announcement. The very early schedule that I sent out had no presentation next week, but we have since added a talk. So there is a seminar next week during spring break for anyone who is around and wants to attend that. That would be great. If you're online and you have questions during today's presentation, you can put them in the chat box. I'll be monitoring that and can let our speaker know if there are any questions. You can also use the Zoom function to raise your hand if you want, and then we can let you or ask you to unmute yourself. That's another option too. So with that, I'd like to introduce today's speaker, Morgan Oneka, who is a graduate student in computational medicine and bioinformatics. Thanks, Morgan.
Thanks for the introduction. And wow, I didn't know spring break was already next week. This semester has been going by really fast. Yeah, so I'm really excited today to talk about some work that I've been doing over the last couple of months, and I'm specifically going to focus on an R package that I've been working on. It's kind of in its infancy, so any questions, suggestions, concerns are very welcome. Feel free to let me know. But yeah, this is something that I'm really excited about and excited to share. So before we go. Yeah. Before I get too far into like what I've actually been working on, I want to take a step back and talk a bit about networks. I feel like sometimes I feel like everyone should probably know at least a little bit about networks, but just as like an overview, because I work with this a lot, I want to make sure we all have the same vocabulary. A network is a mathematical structure that is used to describe relationships between different entities, to describe like a system of things interacting with one another. It consists of two different types of things. We have nodes and edges. So the right-hand side, I have a figure showing what a network is usually drawn to look like. This is a representation that most mathematicians have kind of agreed is the best way to represent it. We have these circles, and these are our nodes, and they represent some kind of object that we are trying to model the interactions between. And then the lines in between the nodes are called edges, and they represent a relationship between the two objects. So an example of this that we might be familiar with is like a social network. We could consider this a representation of a group of people on Facebook, where the individual nodes are people, and then they have an edge if they are friends on Facebook. And this is a network at its most basic. If you are from a more mathematical background, you might be used to calling it a graph. It's basically the same thing. And it can be a little bit more complicated if we want them to be. We can give nodes and edges different features to represent some feature of the object that we think is very important to capture. So we can see this example that I have here. The nodes are two different colors, they're pink and blue. That could represent, I don't know, like gender, whether they correspond to a certain group or not, something along those lines. We can also have edges have different features. We could assign an edge weight. If we're keeping on with the Facebook example, it could be how long they've been friends on Facebook, something along those lines. So that's what a network is in general. And it's something that's very useful in biology, because biology, specifically systems biology, involves the study of a lot of objects that interact with one another. When we were children, most of us probably learned about the food web. As a brief overview, it describes animals eating one another for energy, the circle of life, if you will. This can be represented as a network where we have a node representing our animals that are interacting with one another in that way. We have like a mountain lion and a snake, and then we have like our grass and stuff. So that would be our nodes, would be the living creatures, and then our edges would be this consumption interaction. And this is an example of a directed graph where the relationship isn't the same both ways. So the shrew is being eaten by the mountain lion, but the mountain lion is not being eaten by the shrew. My library that I'm going to talk about doesn't really deal with directed graphs yet, but I do want to bring up that that's a thing that exists and is forthcoming in this thing I'm going to talk about. And then a little bit more pertinent to what I work on the right-hand side, I have a protein-protein interaction network. So in this case, our network has nodes representing proteins that interact with one another to perform some kind of functionality within the living tissue. And then they have edges if they interact to some extent. And in this case, we can see that we have weighted edges. So we have really thick lines between some of our nodes, and then some of our nodes have these like dashed, very light lines. And the thickness and the strength of the lines indicate how strong the interaction between those two proteins is. And then we also have like node features. We have these colors, which I'm assuming correspond with some kind of pathway, because that's usually what these graphs do. So yeah, networks are very important in biology. They are used to model a lot of different systems, whether that's on like a grand, like macro scale, like the food web, or the microscale, like a protein-protein interaction network or a gene co-expression network. So it's very important to come up with a good way to visualize them because they are like a concept, they're an idea. We can't actually like look at a network and see that it exists. It's like a way to represent something more abstract. So visualization is something that is very, very important. Unfortunately, it's also not a really new field. Networks have existed since long before, but as a concept, as a mathematical concept, have really existed since before like computers and all this cool visualization stuff came out. So there are tons of libraries. If you want to look at one individual network, there's so many different ways that you can do it. Any language you want to. I have a couple of examples here that I work with. iGraph is an R package that I find just absolutely essential to my like daily workflow. It's a way to create like graph objects within R, and you can perform a lot of different graph theory functionality on it. And it has what I consider a really good first step sort of visualization tool. As you can see in this upper left, it's it's not super pretty, but it gets the job done if you just kind of want to see, okay, what does my data look like at a bigger scale. And then on the top right, I have an example plot from ggnet. ggnet is also an R package. It's built on top of like ggplot and all of that. It's also really useful for creating visualizations, and they are a lot prettier. The one on the top right doesn't take that many lines of code to make it look nice. The default is just very pretty, like ggplot is in general. It's a lot more useful for showing what our networks look like for I've used this for like presentations for collaborators and figures for papers and things like that. And then on the bottom is something called Cytoscape. Cytoscape is a really powerful, I believe it's web-based application that gives you a lot more control over how your graphs look. It's a lot easier to assign like graph or node colors and edge weights and things like that. It's also really helpful for very large networks because it's a lot more interactive. You can zoom in and zoom out. And scroll around and things like that. So it's really great for like really large gene regulatory networks and things like that. And so if you would just want to look at one network at a time, these tools are very, very powerful and very wonderful and work amazingly. But a problem that I have run into is that I like to compare my networks. I'll create, for example, a gene co-expression network for maybe two different conditions in a control group. And that's where I tend to run into a lot of issues in terms of visualization because these tools are not built with comparison in mind. They want to show one network, and in most cases, that's plenty. That's what most people do. But for me, I run into issues with node layouts specifically being an issue. So what I like to do is like I have a node and it's in the same location for if I have a node representing a certain gene and it's in the same location for all of the different samples that I'm looking at, and that's the ideal. But that doesn't really happen if we're looking at like a random layout. iGraphs plotting tends to be more random in its assignment. And then tools like Cytoscape, they tend to cluster nodes together based on how strong like the genes co-express, for example. So they use edge weight to determine where your node is placed. And that doesn't work if you're trying to compare networks over where we expect the co-expression to be different. I mean, it gives you some source of information. You can see where the clusters are, but then it's like if I wanted to look just at gene one and its relationship, that doesn't really help me as much. And then another issue that I've run into with node layout is that some of the software I use, if a node doesn't have any edges, it doesn't get output from the software. Obviously, if you're doing a comparative analysis, you want to look at the same like gene set, for example, for all of the samples. But then if your software only outputs significant interactions and excludes nodes that aren't important, I run into issues with the graphs not having the same node set, and then everything's laid out differently. Even if you use iGraph allows you to select some preset examples of layouts with like the sphere and the circle, and that gives you some continuity. But then if you have different node sets, it doesn't work as well. And then finally, another issue that I ran into, which was kind of the straw that broke the camel's back for me, was visualization of loops. I find ggnet to be a really good kind of common ground between easy to use but still looking really nice. But it doesn't allow at this time visualization of like loops. And in certain contexts like a gene co-expression network, you don't necessarily expect there to be a node having a relationship with itself. But in some situations, which I'll talk about later, that does happen. And yeah, ggnet and a lot of other similar things, other similar libraries don't include that functionality. So that was the main kind of issue that I was running into when I was trying to come up with figures for my collaborators and their data. And then another issue that I encountered later on was how to more easily visualize topological differences. So like graph topology basically just refers to like the connections between the nodes. We can see that the set of graphs we have. Some that have edges between the red and the yellow nodes, and some that do not. And so if I wanted to, let's say, see how many edges existed between red and yellow, or what patients had an edge between red and yellow, that would be pretty easy to do with iGraph. I could just go through, use some like fancy LFI magic, and that would be pretty simple. But then, first of all, it's not really super easy to visualize. It's just a bunch of numbers. And then secondly, in a lot of the data that I work with, the edges have different edge classes or different types of like that. Edges don't all mean the same thing. So if I wanted to count the number of like pink edges and black edges and then no edges, again, this is something that I can do with like iGraph objects or something along those lines, but it's a bunch of numbers. It's really hard to visualize, and it's really like you want to have a nice visualization if you want to actually talk about this and show like your collaborators and people who are reading your papers like what you're actually talking about, because you can only throw so many numbers at people before their eyes just kind of glaze over. That was another thing that I found lacking in terms of like comparison between networks. And so that is where my visualization package that I've been working on comes in. It's called Differential Network Visualization in R, or DINER, because what's the point of having an R package if you don't have a fun acronym for it? So it's not, I'm not out here saying it's like a super groundbreaking tool or anything like that. It's something that I created to solve problems that I was facing and that I think are very niche problems, but I'm hopeful that this can be applicable to some other people's data. Mostly this was me like trying to use some of the really great tools that exist on top of ggplot, such as ggnet, and then just running into issues with like, especially like the lack of support for loops. So I'm gonna take the next couple of slides to talk about like a broad overview of what this package does. So it is an R library built on top of ggplot2. The number one feature that I wanted to make sure I included was stable node placement across a set of a lot of different networks. So if we're doing a gene co-expression network, gene A is going to be in the same location no matter what, like which individual we're plotting. I wanted to include support for looped edges because that was the major issue I was running into. It also allows us to visualize edge classes and then weights rather easily. And then I also included some functionality for other plots that are useful for summarization and comparison across different groups. So the only required data that you need for DINER are these like node adjacency lists. And what this essentially looks like is if we have, let's consider this a data frame, we have two columns and each row represents an edge in your network. So, and then we have like the source node and then like the destination node, if you want to call it that. Basically, we're saying there's an edge between gene A and B, C and D, C and E in this patient's, we don't want to call this like a gene co-expression network. And then basically all you have to do is like read it in or like create like a list of these data frames and then send it into the function to create like a DINER object. If you have additional information about like maybe these edges have different classes or they have different weights, if you have information about like your individuals, so maybe we can say that like this patient has cancer and this patient's healthy and this patient's healthy. You can include that as well. I have functionality for that, but at the main core, this is all you really need are these like node adjacency lists. So essentially how DINER works is that it creates a node set, which is basically the union of all the input graphs nodes, and then assigns each node to an XY coordinate that is the same for all the samples across all visualizations, so that when we call any of the plotting functions that I've created, it looks at that XY coordinate list and makes sure that all of the nodes are in the same location, so that it's easier to look at these graphs and actually like compare them. So each, yeah.
Just really quickly, there's been some requests to maybe slow down a little bit. I think particularly with the mascot, it's a little hard to understand.
Okay, yeah, sorry about that.
I don't know.
Yeah, okay. So yeah, so this, yeah, so they're like, yeah, okay, so we have like this coordinate list, and that is how we can see where the nodes are supposed to be for each time we try to call like a plotting function. So that's included in like basically we create like a DINER object. I was kind of modeling it off of Seurat a little bit, where you have like just one centralized object that has all the information that you need. That's one of the things that is included. And then we also have like our adjacency list for each sample. So like basically these data frames that I talked about earlier, we keep that so that we can refer back to them. And then so that's what's included when you just call first, like creating this object, and then there are additional functions you can call to assign like node and edge features or like individual like sample features, and that gets saved as well. So that is just a high level overview. And since I was talking kind of fast, if anyone wants to stop me for questions about if you're confused, like why we would want to do this or anything like that, feel free to. Otherwise, I have some sample data that I'm going to go through, talk a bit about like specifically what kind of analyses I do and how I find it useful in my research, and show you basically like what the plot list would look like. Okay. If no one has questions, I'm going to keep going. So I work with spatial biological data. Basically, my work focuses on how can we identify like spatial trends in biological data. I work with spatial transcriptomics and proteomics data. And for this example, I'm going to go through some spatial proteomics data using the Codex pipeline. So essentially how spatial proteomics with Codex works is we have these, we can visualize where the cells are in the tissue. And then we can also I believe this is kind of similar to like multiplex immunohistochemistry, where we can see the like protein expression at like in what location that protein is being expressed at. And so we can get this idea of like where the cells are located. So we get like the XY coordinates for the cell centroids. And then we can also get their protein expression levels. So this allows us to label cells as different types. And with this data set, they labeled their cells as one of 29 different types. I didn't look at all 29 for the downstream analysis because that would be quite a bit. And some of the cell types are just not that interesting. But essentially, we have this notion of where the cells are located within the tissue, and we have what type of cell that they are. And so for this specific data set, it's for patients with colorectal cancer. There's helping cancers tissue for 35 patients. Not all patients have both types of tissue. But the key thing here is that the patients are split into two different groups based on basically how their cancer presents. There's like a DLL response and a Crohn's-like response. And I'm not quite sure what that means in the context of colorectal cancer, but just know that there are like two different groups and they're experiencing two different basically like symptoms and reactions to their cancer. And you might be asking, okay, where does the network come into this? That's a bunch of like spatial data. That's like more of a GIS problem. And that is one way to go with this. But one thing that tends to be really interesting when we're looking at cancer is looking at cell pairs that tend to interact with one another. So cancer is driven by a lot of interactions in the tumor microenvironment. And this is obviously true of like all functionality within our cells and within our tissues. But the tumor microenvironment is especially very interesting because we have these cells that maybe aren't acting normally. We have a lot of immune activity going on in there. As we can see on this like left-hand figure, this is just kind of an example of all the different interactions that we might be seeing going on. And again, since we have cell proximity information, we have this like spatial location of cells, we can kind of infer that cells that are close together in our spatial tissue are likely interacting. This isn't something we know 100% for sure, but we can kind of guess like if they're close together, they're probably interacting with one another. And so we can get this kind of notion of what cells are interacting with one another. Like we can look at our spatial data and see, okay, maybe tumor cells are really close to neutrophils a lot of the time. So we can guess that neutrophils and tumor cells are interacting a lot of the time. And that's something that we can visually see looking at the tissue, but it's kind of difficult to quantify that. But just by looking at it, it's obviously a lot better to have like a mathematical representation. So one thing that I tend to look at with my research is cell pair enrichment. And so what I mean by cell pair enrichment is kind of what I was talking about just now, where two cell types tend to be close together or far apart more than we would expect. So if we mathematically see through whatever means that two cells tend to be really close together, more often than we would expect from random, then we say that that cell pair is enriched. So if tumor cells and neutrophils are close together based on whatever metric, more than we would expect from random, then they're enriched. And then on the flip side, if they are not close together as much as we would expect from random, then we would say that that cell pair is depleted. So if like tumor cells and T cells are like, we have this expectation of how much we expect them to be close together, expect them to be neighbors, and they're not, then that is a depleted interaction. So to get this information, we start off with a cell proximity network. So we start off with a network, but this isn't the one that we're going to visualize later. Basically, every node is a cell, and we connect every cell to its nearest k neighbors based on our spatial information from like the codex pipeline in this example. With some like cutoffs, we can see like this cell right here doesn't have any neighbors because it's really far away from everybody. But we start off by just creating this like network where the cells are connected to their closest neighbors, which are the cells that they're most likely to be interacting with. And so we can see that all of these cells have like have some kind of like label. They're different colors. We've got like yellow and pink and green. And that corresponds to the cell type, and that's our cell label that we're looking at. So let's say that we want to see if our yellow versus red interaction is enriched or depleted. We can say like maybe the yellow is the tumor and the red is the neutrophil. So what we do is we randomly shuffle our cell proximity network, the node labels of that graph many times to create a simulated distribution. So we take our original graph and we change all the colors so that the number of cells that are each color stays the same, but each cell is randomly assigned to a different color. You can see over here, we still have like 2 blue, 1 red, 1 green, 1 yellow, but they're all, or most of them are assigned to different cells. And then we count how many edges exist between our red and our yellow cells. In this case, there's 0. And so what we do is we do this many, many times and come up with this like distribution of how many times we observe yellow-red edges. And so we compare the actual number of edges between those 2 cells to this distribution. And if it's significantly large, then that cell pair is enriched. And if it's significantly small, then it's a depleted cell pair interaction. And so this is the output of what this would look like. I've included a kind of baseline iGraph plot just to show what this looks like before we get into like actually how the package works. But essentially every node corresponds to a cell type. There's 1 node per cell type, and we have edges between cell types that have a significant interaction. So either they're depleted or enriched. We can't really tell from this basic graph yet, but we have some kind of important interaction between those 2 cell types. And then we have an enrichment score that could be used as like an edge weight. And so this is essentially what we are trying to visualize here, because this is helpful a little bit. For this specific data, there's a lot of edges, so this isn't the most helpful. And obviously we can't see like the edge weights or the edge colors. I know that you can do edge colors. I'm not sure about edge weights. I'm pretty sure you can in iGraph, but it was going to take a lot of extra effort. So now let's take a look at how this looks with like my visualization library. So this is what you get for an output from like the Diner's individual network plot function. We can see that we have our nodes and they're all spread out. We have our different edges, but now they have colors and like a gradient attached to them. So let's see, we have like our red edges indicating like an enriched interaction, meaning that like CD4 positive T cells are close together with Tregs more often than we would expect from random. Then we also have these blue lines, which indicate a depleted interaction. So Tregs are near one another less often than we would expect from random. And creating this with the library that I built didn't take too many steps. It was about 5 steps. So I read in my output from the cell pair interaction library that I use. I subset just the node adjacency rows, the node adjacency columns, and then I created my Diner object. I attached the edge weight and the edge color information using 2 different functions. And then I called this this function, which is plot individual. And it was a pretty simple process, but maybe I'm biased because I wrote the code. So yeah, this is what one graph for one person looks like. One thing I do want to point out is that you will notice some nodes that don't have any edges. With most graphing software that I run into, if a node doesn't have any edges, it deletes it from the visualization. You have to like tell it, oh, like keep these in, and sometimes there's no option to do that. Basically what this means is that this individual patient doesn't have any significant interactions with like the CD68 positive macrophages or the CD11B positive monocytes, but some other patient does in this group, and there are like 50 individual samples. So This is important down the line, and it's important that we don't have edges here for when we're comparing to some of these individuals who do have edges out of those nodes. So anyway, the whole point of this is to do like these comparisons between individuals. That was the whole like purpose of creating this. I'm just going to go through a bit of like how I would talk about this with like a collaborator. So we can see like patient 1, we have this like interaction between Tregs and CD4 positive T cells that is enriched, and it's pretty highly enriched based on the color. But then we go over to patient 2, and we can pretty easily see without having to dig too much into these plots are a little bit messy, and we're going to talk about how to clean them up in a minute. But we can see that this edge over here is blue, and it's like a light blue, so it's pretty significantly depleted in this patient. So we can see that these two cell types, they are both very important and have important interactions in the 2 different patients, but like for one patient, it's very likely that they are interacting, whereas with patient 2, they're very likely not to be interacting. And then a second thing that we can notice is that, again, patient 1 didn't have any macrophage, CD68 positive macrophage interactions that were important, but patient 2 had quite a large number of them. We can see a lot of edges going out of this node. So yeah, this is basically the kind of analysis that I would do in like a meeting with a collaborator, or like for a grant application, for example, just kind of pointing out maybe patient 1 and 2 are representative of different groups. And so we can say, okay, we see these key differences between these 2 groups. So yeah, that is the basic usage of these comparison plots. But again, they are kind of messy. This data had a lot of edges coming out of the nodes. So there, I included a feature that allows you to subset based on like one node of interest. So for this example, I looked specifically at Tregs. Tregs are regulatory T cells. They're usually pretty important in cancer. So I specifically subset based on Tregs. And so we can see that only the edges that are connected to Tregs have the actual colors and like opacities for their edges. And the rest are still there. I didn't want to like completely delete them because I would hate for somebody to just like be half tuned into a presentation or like it's just mindlessly scrolling through my like paper on the internet or something and like look at this and say, oh, Tregs are the only ones that are important for this patient, without like reading the figure legend or something. So they're still there. They're just the kind of really like gray in color so that you know that there are some interactions between other cell types. We didn't forget about that, but that we're focusing in just specifically on the Tregs at this point in time.
Hey Morgan, a question came in.
Okay.
Before you get too much further. Dana asked, sorry if I missed this in the single patient plot, but can you explain the enrichment depletion versus positive negative values within the enrichment depletion?
Oh yeah, okay. That is a good question that I did not go into. So thank you for pointing that out. So I can see how that could be confusing. So yeah, so with the depleted versus enrichment, we have basically, yeah, so essentially like 0 means that there's like no significant interaction. I feel like maybe the scales got screwed up or something. But yeah, basically, like a 0 would indicate that there's no interaction, and then like a +1 for enrichment would indicate that there is a really strong enrichment. And then with depleted, it's the opposite, where 0 means, well, 0 still means that there's no interaction, but the -1 would be where we'd want to go for a very strongly depleted interaction. I do think the labels got kind of screwed up because there shouldn't be like a 1 for positive or a 1 for like a depleted weight. Does that make sense? So I do feel like the labels got screwed up, but they are supposed to be like 0 to some negative number for depleted, and then 0 to like 1 for, or 0 to some positive number for enrichment, if that makes sense.
So Dana followed up is confirming that you shouldn't see 1 for depleted and then not see -1 for enrichment.
Right. I do think that the labels got a little bit messed up when I thought of this, but yeah.
Thank you. There's another question that came in from Anthony. For the plots, how do you determine layering of edges? For example, would the greatest magnitude of the edges be shown in front, lower changes of weights, or would it depend on some factor?
Oh, okay. So yeah, that is a good question. So the edges are plotted in order of it's not based on any kind of like weight. So it's if you look kind of closely, you can see the blue ones are beneath the red ones. So essentially what it does is the plotting function cycles through all of the different classes that it sees for edge types and it really just goes to whichever one it sees first and then plots that. So I might alphabetize it if I remember correctly, but essentially it plotted the depleted edges first. It plotted all the blue edges, and then it plotted all the red on top. And so they are, at least on the left, for the overall like network plots, there is like a lower opacity. I think it's like 80% opacity, so that you can see beneath more clearly, because it's not like you can really control. I guess you could control which one gets plotted first. That answers your question.
Yep, it does.
Okay, awesome. Okay, so that is the overall. That was the first like motivation that I had when I was creating this package that I'm working on. But then my advisor came up with a really good idea, which is this concept of like a consensus network. And it wasn't necessarily like he came up with the idea, but like he said, this would be something really cool to include. So what a consensus network does is it counts the number of edges that exist between any two nodes for all of the networks. So for this one, we have a little bit over 50 individuals that are like within our sample. And so what it does is it goes through every single patient's network, and it counts if it has an edge between that pair of nodes or not. So we can see that there are some edges, and again, this is a very messy plot that we can subset to look specifically at one like target node. But we can see that there are some edges that are a very dark green, indicating that most, if not all, patients have a significant interaction with that pair of cell types. So we can see that like vascular cells versus themselves, like the vascular cells tend to cluster together very frequently, and that makes a lot of sense because we get like vasculature. And then we can see maybe some of these others. We see most of the edges coming out of this T cell group are like very light, so there aren't that many patients that have that interaction type. And so this kind of gives us a sense of like what cell pair interactions are very, very important, to the point where like maybe they're not even that interesting. Like if we see that Tregs interact with one another in every single patient, that maybe isn't something we care about so much if we're trying to compare groups. But some of these lesser occurring ones are kind of interesting because that indicates like maybe only a subset of people have them, so that could be something worth like investigating further. And like these really strongly occurring ones could also be very interesting if it only exists like within this cancer type versus like another. But yeah, so that's what a consensus network does. It kind of just gives us a sense of what interactions might be worth following up on. It doesn't tell us a ton like right here, right now, but it gives us an idea of like maybe what we could look at in the future. And one thing to note is that right now it is counting all edges. It's not counting like depleted versus enriched. That is something that I am working on and should have working in the next couple of days, honestly. But that is something that you can also kind of hack your way into doing now. If you create just like two different diner objects, one for just the depleted edges and one for just the enriched edges, and then you can create like two different consensus networks if that's something that you're interested in. But hopefully I'll have that up and working soon. But you can subset based on like the group that the individuals are in. So this is comparing group 1 on the left and then group 2 on the right. And I pointed out earlier that interesting GATA3 positive T cell set of interactions was not occurring in a lot of different patients. And what I found really interesting when I was looking at this was that this specific interaction only existed in patients that were in this first group. None of the patients in the second group had any edges coming out of that cell. So I thought that was pretty interesting. That is something, this is like open source data, so I'm not really doing hardcore research with that. But if I were to find this in like a collaborator's data set, I'd be pretty interested in. That'd be something we'd want to talk about and include in like a paper or a grad application or something. So that, if you do want to look at edge class comparison, I do have this pie chart visualization for that. So what this does is it looks at every single cell pair and it calculates the proportion of edges that belong to each class. So it'll go through every single patient once again and count the number of edges that are depleted or enriched, and then it assigns everything else to like a third class. So if a patient doesn't have an edge between those two nodes, it just considers it like other or neither or whatever. And what I find this really helpful for doing is looking at what cell types are more important, because it can be kind of messy to look at these consensus networks when there's like a lot of cells. You can kind of see like which ones are more important, but I find this to be a little bit more interesting, especially because you can include the like edge class as well. So we can again look at like the GATA3 positive T cells, and we can see that like all of its pie charts are mostly gray. So for the most part, like these cells are not super important. There aren't that many important interactions, including those cell types, which is something that's kind of interesting, again, to look at patients who do have the interesting interactions. But then we can scroll down a little bit further. This row shows all the interactions with tumor cells. And we can see that a lot of patients have like depleted interactions with tumor cells, which is kind of interesting since most of the other cell types are immune cells. So this kind of shows us that for a lot of patients, they have like tumor cells staying really far away from our immune cells. Something interesting to look into, but then I was like tumor cells are enriched with themselves, which is pretty much to be expected. Yeah. That's all I want to say about this one. Yeah. Okay, so I have a similar visualization that that last one is kind of good for getting a high level overview, but if you want to narrow in on a specific interaction, those pie charts are kind of small. They take up a lot of extra space because they're diagonal, they're like diagonal symmetric. So what this pie chart visualization does is group-wise comparison of basically the same exact information about like interaction type. What how many patients in a certain group have either like a depletion or an enrichment for that interaction. Basically answers the question like, does the frequency of edge class differ between groups for each interaction that we're interested in? So one thing that I noticed that was very interesting right off the bat was this like adipocytes interaction. A very large proportion of patients in group 1 had an enrichment in that cell type interaction, meaning that the adipocytes were really clustered together for those patients. Whereas if we looked at group 2, there wasn't a clear preference between depletion or enrichment, which is kind of interesting to think about. This would be something that I would want to look at a little bit more with maybe some other data or some further like downstream analysis. And then we can also see that for a lot of these interactions, like there are that significant of differences between the two groups, particularly like the adipocytes versus B cells. This interaction was almost the exact same distribution among the two groups, so we can pretty much rule that interaction out as being interesting for comparing the two groups. So basically the whole idea is like, what kind of interactions do we think might be driving the differences? So we can see that like maybe adipocytes versus themselves is a good candidate to look at further. Maybe this natural killer cells as well would be interesting because there's a pretty high depletion in group 1, but not in group 2. Something to look into. Basically the whole purpose of this visualization and the ones before it were really just like, what differences can we see, and then we can do further analysis down the line to see like why might these be different. And yeah, so you can see that there's only a couple of cell pairs listed on the y-axis because if you run it on everything, it looks like this. That is something on my to-do list to make this like a more columned, like logical approach. But for now, you can just input like a list of what pairs you're interested in. Your cell type 1, cell type 2 with a slash and 2 spaces in between. And then you can get a much more cleaner version if you want to. And so that is basically what I have so far for Diner, the first R package I've ever really made in my life. So basically, to summarize, it's a network visualization with comparison in mind. There's some additional functionality for further comparison, whether you want to look at the prevalence of edges between a pair of nodes, looking at like edge class composition, and then specifically comparing among different groups. And my example looked at like a cell pair interaction enrichment network, but this could be useful for other applications as well. I've used it a little bit with small subsets of gene co-expression networks. If you have like a lot of edges and a lot of nodes, it gets kind of messy. But for like a small like community within a gene co-expression network, it's also kind of useful for just kind of looking at that kind of stuff. So currently it's not like an official CRAN package. It's still in a very, like I mentioned, it's in its infancy. It's in a very like alpha stage right now. But it is on GitHub. I have the link at the bottom. All you have to do is like copy the R file into like, your project directory and then call it using the source function. And I do have an R Markdown tutorial provided that goes through like the specific commands that you need to run all the functionality I talked about today. And then some future work. I've mentioned a couple of different things that I want to include in the future during my presentation, but I also wanted to include additional support for node classes. So right now, if you think that like all of the nodes were colored by like what cell type they were, but sometimes it's interesting to look at like tumor versus immune cells. So like coloring all the spells that are new, like the T-regs and the neutrophils and all that one color. That's not something I've done for any of my like collaborators yet. So I haven't included functionality for that, but that's certainly something that I want to look forward to for the future. And then I also want to include some further integration with iGraph. So in some of my other work, I've been talking a lot about like hub scores and things like that that you can implement with iGraph. And I'm working on a way to kind of include some visualizations about like hub scores and stuff in my library. But right now, that is still in progress. And so yeah, that is my presentation. That's what I've been working on for the last couple of months. Thanks to everybody who asked all your insightful questions, and I'm happy to answer any more questions if you have them.
Thank you, Morgan. If anyone has questions, you can put them in the chat box, or you can use the Zoom functions to raise your hand or just unmute yourself. Give it a few minutes in case anybody wants to take their time typing their question. Not seeing any questions at the moment or hearing anybody trying to ask me questions. So I think maybe most of the questions were asked during the presentation. So I'll thank Morgan one more time with a round of applause. Thank you. Next week, we'll be learning about bioinformatics, a multi-way interaction in the genome. So hopefully everyone can join us next week. Oh, wait, hold on. We're not done yet. Someone is in the process of typing a question. So
Okay, awesome.
I'll finish up hopefully people will join next week, but we do have a question coming in, so we will continue the presentation. Wait for that question. Thanks, Dana, for letting me know that you were in process. I never know how long it takes.
Yeah, it depends on how like complicated the question is too.
Yeah.
How long it
It's
takes
like
to
that
get that.
awkward silence where I'm like, I feel like I should give you more time. But
Yeah,
I wish Zoom would tell me when someone's typing. I feel like it used to do that, but it doesn't anymore.
yeah. So many other things to do, like Discord does. And
Yeah.
and Slack does too.
Like you can see, that'd be really helpful.
Yeah, that'd be super helpful, or at least having the option to turn it on.
Yeah.
If it is, I haven't seen it.
All right. Dana's question is. Now I know why I took her so long. All right. It's a really nice talk and interesting tool. For your example data, it seems like the spatial data was at or close to single cell resolution. However, much spatial data is not a single cell resolution. How would you treat a data set where the data is likely to be an average of several cells and classification of the cell type might be more difficult? Is it up to the researcher to classify and determine the relevant interaction?
That is a good question. I'm trying to think. I feel like it would depend on your data. What I would probably do is if I go back so this is definitely something that has been addressed at some point. So I can like that far. Okay, so what I would do is when we're creating these like nearest network graphs. So I'm thinking about like the 10x like Visium platform, where like they're all like equally spaced. And so for that, I would just connect to all the adjacent, like you get that kind of like honeycomb structure where like they're like on a diagonal from one another and you have like 6 neighbors. I would just I would do this exact same thing and then just connect to all of the like ones that are adjacent. I think that like half answers your question because there are still some other methods that aren't like regular, but yeah, I don't know. It's definitely something to think about. I think it's, yeah, that's how I would solve it. But I think it is also like up to the researcher and like, I feel like knowing that the whole idea is we want to capture like if the cells are interacting versus not. And I think like depending on your platform for getting with your spatial and your expression information, like I think you have to like look at see like what would constitute like spatially proximal enough to be interacting, I guess, if that helps. But it does depend on your data, I would guess.
Your reply that that's helpful.
Okay, good, glad that was helpful. That's a really good question. Definitely something to keep in mind when we're doing this.